Okay, so let's just dive into it. Thanks for everyone for another week with us. I always put this up for an in-person meeting. No luck there, but and no new papers that I know of. If I missed any papers, let me know. I usually have this slide here just in case people want to check out any of the papers people have put out in the group. Um, yep, and then just the basic updates. As I mentioned every week, Al has got a group of people together to work on this nice case report for long COVID. Anyone interested to help out? There's a lot of data that he's gathered from this patient that has agreed to be, have us to look at it, and he has done all the proper legwork for this. So if people are interested, there's definitely more the merrier, could help, be great. Okay, let's go into the news. Was debating whether I should discuss this latest conspiracy theory about where COVID started, so I figure why not start there. I think this AP article kind of described it the best, where, as most of you heard, there was that Energy Department announcement, or they didn't reveal what was there, but they said there could be a potential, again, going back to the lab leak, saying it could be someone who might have been infected and then transferred it. Now, what most people in the media have ignored, saying this is with low confidence, that's why I highlighted here, was what they mentioned. And also, a lot of the theories that, if the theory is more outlandish than, reality, I think it's probably best to think of it as a conspiracy and not a real thing. So the science backed up. And this article actually does a good job going over it. And, interviews, for example, this virologist that she says, the vast majority of evidence continues to support natural evidence, but as a scientist, of course, we can't rule out anything. So once new reports come out, sure, we'll look it over, people look it over, and then come up with the conclusions that would be proper. But, as this morning, like the FBI director said, that possibly there, not necessarily jump to conclusions like all that we hear in media. But that's it. I just wanted to bring that up. I'm sure people have been reading about it. But again, the Energy Department clearly said it was like with low confidence that the virus escaped from a lab. But there's reports that make it public. Okay, so now to the actual science. There's this paper.
So is this one of those places where actually covert should maybe make a statement, do you think? I mean,
Yeah.
I have some troubles with this. I mean,
Yeah.
there is evidence, the fact that they say that they found SARS or some sort of SARS-like virus in bats that was so many miles away from the Wuhan Institute of Virology. I mean, the fact of the matter is the first cases cluster off the seafood market or the live market.
Right.
But I think many people are just looking at headlines and what's being selectively reported, as opposed to, the reality is you cannot rule it out as being as a potential that it came from the Virology Institute. But the fact of the matter is the first cases came from around the market. And second,
Right.
is evidence that suggests that SARS-CoV-2 virus has been detected in samples from around the world earlier than December.
Right.
So
Yeah.
it just requires, I think, more nuanced reporting than this. The conspiratorial nature in the way that this report actually was released. It just needs like a calm comment, in my opinion.
Yeah, I mean, I agree. I mean, I don't know if people want to write a little note and put it out somewhere. I'll be up for that. Discussing all exactly what you said, Rob, the pros and the cons of what's going around. I don't see why it's no harm, right? It'll cause actually more benefit, right, to get this information out so people can actually read it, make up their own opinion. But at this point, too, the other point is that does it really matter of the origins? I mean, for some people it does, but for me at this point, I'm kind of like, I think the science is probably more important to figure out how to come up with novel therapeutics or ways to stop long COVID. But I mean, both are valid.
You're absolutely right. It's completely the point at the end of the day where it came from. It's scary. Right. We need to deal with it. I think it's more just trying to be more responsible in reporting rather than just trying to get the headline that allows you to get more likes on social media.
Exactly. No, I agree. I agree. Yeah, it's more of an annoyance than anything else for people like us working on it, right? So, yeah, I mean, we could talk offline, or if people want to come up with like a short little commentary. I'd be up for that. Good idea. Okay, there's no other comments, but yeah, I think it's a great idea. Um, on a better news, I mean, this is. Oh, sorry, someone had to say something. Okay, no, okay. On better news, you might have heard about this. There's someone's microphone is hot, so if you mute it, that'd be great. But there's this vaccine that's a nasal, vaccine that's developed by this company, Blue Lake Biotechnology, but also people from Yale that were involved. Probably people recognize Akiko name. She's a prominent COVID researcher from Yale. Many papers by her, but this was a science paper that came out in October last year. It's starting to describe this vaccine. It's an interesting paper. I think I remember seeing it back then, but I didn't. Now I've paid attention to it a little more because of these headlines. But because now it's in clinical trials, or phase 1 clinical trials, I should say. But what it does is take, it uses the spike from the SARS-CoV-2 as a like an mRNA type intranasal vaccine. This is kind of the summary figure about what it does. And so what it does is the whole goal is to use the pre-existing immunity straight from the nose to actually boost the system and target the virus itself. Supposedly, from their trials, they're saying there's 86% more reduction in symptoms than opposed to what we know about the normal booster shots, around 43%. So it seems promising, but it's only in phase 1 clinical trials. But if you want to look in the science paper that goes into the biology behind the actual intranasal vaccine that they're creating, be an alternative. So some other preprints that I saw today. I guess I focus more on long COVID. There's a bunch of long COVID things, like papers I saw on preprints. Again, these are preprints. I didn't see much more for like looking at acute of COVID, but this one's talking about potential biomarker, in the kidney for renalase, which is a kidney releasing biomarker for kidney damage. Again, this paper is sort of you could read it. I mean, there's only a couple of figures, a few figures in there, and they kind of just look at patient data around 437 and then correlate with survival and death related with this marker. So you could take it with a grain of salt, but overall, I guess general theme of the paper is showing that the lower levels of this is related with mortality of COVID. So maybe outcome-wise, this could be a marker to look at, but potentially. And then this other paper here, again, preprint, they're looking at conditions, pulmonary functions. Basically, they looked at EHR data from no molecular data, but they looked at basically, outcome, when people report when they get their pulmonary tests, like, for lung function and things like that, like breath tests and things like that, CO2 concentration. And overall, they see even 12 months after the onset of you getting COVID-19, there's a person getting COVID. Someone's mute. My microphone is hot. So it definitely has a reduction in pulmonary functionality based on the tests that you get in the clinic. Now, some long COVID papers, like this one paper which came out recently. When did this come out? This came out this past week, I think. But they're talking about basically long COVID patients show, it's a very short paper, but it basically looks at lower levels of brain oxygen. This is the one figure and table from the paper, but they did a pretty good job, looking at different images and hemoglobin levels and the concentration. They basically pinpointed this location where it's a lower level of oxygen occurring, and might explain potentially reasons why some people might have decision-making issues. Again, no biology, but just kind of looking at hemoglobin levels in places in the brain. Again, I'll share all the slides with the links here. So this is another paper that was looking at a multi-organ impact based on markers you get from the blood for long COVID. So this is basically looking at, again, EHR data, looking at all the various factors related with organ damage, that you might look at when you go get your yearly checkup. And so they looked at for long COVID, a 1 year study. And these are a couple of figures from the paper where they basically said, definitely, as probably a lot of us here would suspect and know, is that definitely looking at the markers related with COVID, it has big impact even a year after you were infected. And this is particularly looking people like who are reporting long COVID symptoms, also comparing them with people who weren't, the baseline. You could see, looking at different markers related with different organs and conditions, like fatigue, coughing, headaches, and so on. And then, some other markers here. So that was interesting to see. Again, no actual biology or molecular biology looking at it. It's more these kind of studies they did. Now, this paper actually looks at the biology. So I thought this might be interesting for the inflammation group, for their paper, and other folks. I know the senior author, so I'm going to reach out to him, Jacob, to see if he wants to give a talk to our group. But what's interesting here is that they looked at they mainly did mouse studies. This is all mouse studies, no human data. But what they did is they wanted to see if SARS-CoV-2 can cross over the blood-brain barrier, which they show in this paper that indeed it does. I'm going to show you a few figures from this paper. Actually, one figure. These two figures from this paper. And then they use a mouse model that's of Alzheimer's disease and related also with neuroinflammation. So it's probably some of you here who are more familiar with CNS models, maybe Rob Miller and other folks might know this model. But and Jacob's not a virologist; he's more a radiation biologist and a CNS person. So I imagine they're doing some studies and saw this happen and then pursued it pretty nicely. There's a lot of figures in this paper, so I didn't get a chance to look at them in depth, but they did a really comprehensive study in these two mouse models. And then what they did also looked at all of the spike 1 protein variants, and then also figured out how well does it cross the barrier, blood-brain barrier. And then the final, climax of the paper, they're showing how a lot of neuroinflammation is associated with these increases, of the either S1 protein or SARS-CoV-2 that crosses the blood-brain barrier in the different regions of the brain. So this was interesting to see that they looked at different blood-brain or different inflammatory markers related in the brain, like some of these here, for example. But if someone reads this more in depth, yeah, go ahead.
Steve, you may have said it. We'll come back to that question over and over again. So they're picking up the virus. Is it replicating virus or not?
So they did this spike 1 protein variant. They did two models. They did one that was replicating, just the whole, I forget what model they used for that. And then they also did just the spike 1 variant to see if that goes. That part isn't replicating, yeah. But the two other models were.
So again, okay, so what do they conclude? That it's active virus from the conglomerate of what they saw?
Right. Keep in mind, this is like a long COVID mouse model. So it's not so it's not maybe in a human model, it might not do that in long COVID.
Understand, right? Yes, understand. Thank you.
Yeah. But it was interesting. They did a pretty so the two COVID models, SARS-CoV-2, I forget which variants they picked, but the S1, they did, as you can see, all the different variants they went through. And they said all of them do basically to a certain degree, Omicron maybe more than others and Delta more than others, as they mentioned somewhere in the paper, more than other ones. So that was interesting to see in that paper here. And again, I'll share the slides so you can go to the paper and read about it. Now, this paper just came out this past week, 27th, or is it? This past week. So this was pretty interesting, and also this is probably a good source of data because the single-cell RNA-seq that they did. Again, this is a mouse model they looked at. This is a long fibrosis, long COVID kind of mouse model they're using, a humanized model, which I'll get into in a second. So how well does it relate to the human long COVID? I don't know, but I guess people who might be expertise in that. But the data, again, is pretty interesting. So what they did was, they're looking at this long COVID relationship to fibrosis. And I guess people are starting to report that more and more. It could be one of the negative impact of long COVID, that we get early fibrosis occurring in people, lung fibrosis. So what they did, they used a June mouse model. June is probably people from cancer knows June as an oncogene. But also, I guess in the fibrosis field, this mouse model is used to show advanced or a good model to actually show advanced fibrosis occurring. And then, what they did, they looked at, again, this is probably relevant for the inflammation group because they're saying, in long COVID, this innate immune cell activation occurs in the fibrosis model and induced by COVID in a sense. So because they infected the mice with and without COVID. So this is just one of the figures that I thought was interesting here, where they're looking at, this is the first figure. And then this whole point of this figure is saying, indeed, that if you globally looked at this model with the different mice and then clustered the different kind of immune cells together in the single cell data, that innate immune mediators are one of the key players with COVID lung fibrosis. There's probably genes here in their volcano plots that people recognize related to that. And interestingly enough, the little cluster 7 here from their single cell clustering, which I forget what this is related to, but you see oxidative phosphorylation is part of it. So mitochondrial, they don't talk about mitochondria as far as my skimming of the paper went through, but they might. But they mainly looked at the, again, innate immune factors and relationship there. There's some probably immune related genes here that is relevant to our inflammation group. And another figure from this paper. Now here they're looking at innate immune. I put the figure caption here next to the figure here. But this is looking at innate immune infiltrates and fibrosis in mice after transitioning to COVID lung fibrosis. What they're categorizing is that. So they did a treatment with doxycycline for June induction. And this is, I guess, their model to promote or exaggerate or exacerbate, I should say, lung fibrosis. And then they did it with a human ACE2 and S protein compared to that, see what happened. And you can see when they induce lung fibrosis, that obviously survival's a lot worse for the mice. But I mean, what they're looking at here also is that different markers related to this, within the spike or the different inflammatory markers or innate immune, like June, IL-6, CD11B. Again, all this data is available somewhere because they had to deposit it. So if people wanted to mine this data, and it's a mouse model, no human data again. So there's another figure that, I think this is the last figure I'm going to show from this paper, but this is their humanized mouse model, which people are probably familiar with the SCID model, but then they use this knockout IL-2RG. I'm not too familiar with this one, but probably someone else is here. That could speak up, but what they did here was try to see if they could rescue the fibrosis based on what they knew with the innate immune factors throughout the paper that they're showing. So this is the experiments here, and they show that by blocking antibodies against CD47 and IL-6 in the humanized mouse model of the COVID lung fibrosis, that they're able to rescue some of the impact of fibrosis within these, I guess, quote, long COVID fibrosis model.
So, Afshin, quickly, this is incredible overlap with the acute inflammation data that we have. If you could send the paper. What's really remarkable, and we can present this to the group later, and it was data just as of yesterday, I don't think we were on, that we have this finding in the autopsy tissue that there's tremendous potential damage to the mediastinal lymph nodes. And all of this fibrosis and everything, Rob Schwartz just showed us yesterday. The transcriptomics has total correlate with wet bench showing fibroblast buildup, fibrin. But the key is that's all relatively acute in patients. So this begins to join the possibility that that's a carryover into long COVID that has tremendous implications.
I'll send this. I'll send the thing. I'll send the link to the paper too. In this.
Everybody might be interested in this.
Sorry, I missed the meeting yesterday. I was in the lab. But,
that's okay. That's okay.
But I mean, they didn't talk about any other organs other than the lung. So they might have missed the bigger picture that you just mentioned.
The overlap with the mediastinal lymph nodes in the patient autopsy data, especially probably with the most severe patients, bell-shaped curve, is just remarkable for this model, at least, reference to what we're actually seeing in autopsy tissues from patients who are relatively acute in the infection inset that died. So it's amazing.
Curious to know if we reach out to these authors if the mice did save any of the other organs because that might be something useful maybe for us for follow-up. Maybe if they have.
I think the group would benefit. We sure would from doing what you said, having them come in and give a talk.
Yeah, I'll reach out to him. I don't know the authors here, but I'll email them and see the senior author, Geraldine, see if they want to give a talk to the group. But
Thanks.
Yeah. Thanks, Steve. Yeah, definitely. I thought the inflammation group would be interested in this when I saw this paper this morning. It's a pretty long paper, so I didn't get a chance to really look at things in depth. But I figure people can look at it because they looked at a bunch of different factors. There's only the few figures I highlighted, but different innate immune markers and things like that. It's a pretty comprehensive paper they did. But yeah, so I guess the implicate. Yeah, sorry. Go ahead. Was there someone about to say something? No. Okay. Yeah, I mean the implication they're making is that even though it's a mouse model, that definitely the cases that we see or the reports that are coming out that people are having long COVID, fibrosis related long COVID, symptoms, that this could be one of the explanations for it. And this one is this lesser study, but I thought it was interesting. It's a preprint that came out that just was looking at potential treatment that you can do for long COVID patients, and they're saying that they found this group in Denver, University of Denver, that saw potentially that the people who have concussions and they do treatment to concussions can benefit. Long COVID patients might be able to benefit to recover some of their movement and things that related to long COVID that was really similar to maybe concussions. Basically, they noticed that in this preprint that people who had long COVID, not everyone, but some people had motor skill issues with like keeping their focus and things like related similar to concussion. And then when they started doing treatments similar to people who have had concussions, that it improved their outcome actually for their mobility and keeping balance and things like that, and spatial reasoning tests. So it was interesting to see that. Again, no biology, but there's maybe some benefit here preprint. But so take it with a grain of salt, but it's interesting to see if this would hold up.
Were those biochemical treatments or just physical ones?
Physical treatments only. Yeah. Yeah, it's whatever the physical therapy, they would do for concussions. But that's the papers. But so going into what's happening with the variants. In the U.S., as we know, it's XBB.1.5 really taking over. I'll show you in a second with the CDC tracker that indeed, it's always the East Coast that this has started with, and pretty much going over. You'll see in a second. The good news is still the wastewater is showing even gradual decrease, keep decreasing. So that's actually good to hear. But although it's still the cases are in the wastewater is still this is Omicron, pre-Omicron. So it's the spike that you see there is coming there. It's not obviously down to where people want to see it, down to zero or very close to zero, which would be considered I would consider an endemic in my opinion, because I don't think we're there yet. But it's going there for a long time, and we haven't seen any new variants pop up to compete yet. So in the wastewater, you can see here the purple is the XBB, pretty much that all across the nation in the U.S. So that's actually good news to hear. And again, this is the CDC tracker for the variants. You can see definitely in the New England, Northeast area, even just the East Coast area in general. XBB.1.5 is the dominant strain, and then West Coast is taken over, but there's still BQ.1.1 that's there depending on where you are on the West Coast. There's other variants that are very obviously always circulating, but they're very low percentage, and it won't make a difference, I don't think. For the outcompeting XBB.1.5. There's a little feedback. Okay.
I hate to ask. I wanted to ask you something. In your experience now, all of us, is this the first time that at this point, when you look at that bar graph and everything, you don't get a hint of another variant that is progressing and looming and potentially taking over? It just seems.
Yeah. I was on the CDC Spears call earlier this week too, and I'm sure other folks were, and they commented indeed that it's probably. It hasn't been like that. Usually you see another one taking over, like we see in this graph here, right? BQ.1 was there, and then also this one popped up, and then we always see one subsequently before that, like the BA.5. But
right.
So far,
in your wave theory, where you showed those waves,
exactly, and the wastewater is showing this too. Like, this is probably the longest plateau that we're going down. So I don't know what that means. Is is there something that's going to sneak in and take over, or is this, is this it? Is XBB.1.5 is maybe the the variant we settle on, and it'll just be circulating in the population. I
Or
mean,
is
the
it
idea
simply we're not sequencing enough?
that's another that could be, well, yeah, that could be another issue too. Yeah, the sequencing itself, the surveillance everywhere. That's the whole other issue I didn't bring up, but that's very much true. Like, as we know, in the UK they stopped this few 6 months ago. So I don't really, no one can really get reliable data from the UK. I mean, people are still sequencing. There's individual labs that are luckily doing it, so we could get a small proportion of what's occurring. In the US, still the labs are sequencing pretty well, but by May that's going to change. So I think we're still pretty good with the sequencing depth, and I mean it's dropped tremendously since last year, obviously since the past 6 months. But I think we're still getting a good in my opinion, I think we're getting a good representation of the variants based on the sequencing done. Now, when we roll over May, that might be a whole different story with everything, or even in the next couple of months, because there's a lot of labs that are just starting to shut that part down for the tracking and sequencing in the U.S. But I mean, to your question, Steve, it could just be a lack of sequencing, or it really is maybe XBB.1.5 outcompeted all the different variants, and it's just being the dominant one now. I don't know, but I mean, it certainly looks like that based on the wastewater and this. But Rob could be very much right too. Globally, I mean, everywhere is kind of the same. There's some countries that are like, just as Germany, I show you, it has a little spike up and then goes back down for hospital admissions. But the case numbers are not very reliable anymore to report, as just like the sequencing, we know the cases are reporting are becoming less and less frequent or reliable, because there's been a lot of the places don't report anymore. But hospital admissions might be a way to look at it, or ICU, and new deaths. Although the deaths are not representative anymore, I think, of the cases, since the vaccines have reduced that tremendously. But globally, we see that this is kind of more of everywhere looking into it. But this is just kind of a more, I guess, macro level view. So he's looking at Europe and different places, and you can see everywhere is pretty much going down. This was the spike in Hong Kong that mirrored a lot of the Asian countries that we saw earlier this year, or end of last year and earlier this year. If you look at Europe, France and other European countries kind of mirror what's happening. Again, case numbers not so well reported anywhere anymore. So, but hospital admissions, I think, are somewhat reported accurately still. I mean, Germany has a slight rise in cases. Again, might be a lot higher than this because of the lack of case report, but you can see there was a rise in hospital admission, then going back down. U.S., as we see, this pretty much mirrors the wastewater. There's a slight echo. I think. Okay. Yeah. So then, but that's why I would typically look at the wastewater, because that luckily is being reported, and that hasn't been gone down. Biobot hasn't really gone down reporting wise, or sampling wise, I should say. So that mirrors what we're actually seeing with the cases in the U.S. South America, Brazil, same thing as everywhere else. So hopefully the reporting there is still somewhat accurate, or as accurate as everywhere else, I should say. And then in Asian countries like Japan, I think mirrors pretty well other places like Singapore and Korea. They've all kind of mirrored each other fairly well. You can see this was the spike that they had with Omicron and the last wave of Omicron. Something I didn't have time to show, but what we learned from the CDC Spheres meeting earlier this week, people who attended that, is that I think a lot of the other countries like Asia, there's still the other variant of Omicron there. I forget which one it was. Maybe someone remembers. Maybe someone, but it's one of the other variants of Omicron. I think is BA.2.75. I could be wrong. Don't quote me on that. But, you know, it just shows that everywhere sort of is still stuck on Omicron, which might be a good thing. I don't know. But all the cases everywhere is going down. But that's it. I mean, I don't know if people have any questions, comments, things I didn't bring up that were important to bring up that I didn't talk about.